Hello, this is Umar here. We will discuss about the coronavirus infection. So we will discuss about the physiological aspect. So here is the SARS COVID-2 is the major example for the using of this infection, while the COVID-19 is under the study. So the COVID-19 is contain a ACE2 spike protein. So that is ACE2 spike protein, and there are a lot of family of this coronavirus classification which that is under the study. So basically this structure is like that, and the spike protein is useful for the infection site. So that is infected area into the lung epithelial cell. So we will discuss also. So here is the trachea, which that is allowed. Trachea contain a ciliary cell which that is used for the trapping of dust and particle, while the lungs contain a basically here is a trachea which that is ultimately branched in divide into the branches which that is known as the trachea, and this trachea convert alveoli, and this alveoli made up of basically alveoli is the plural, and the alveolus is the basically is the single alveolus. So that is contain air sac is also alveolar sac, it is also known as. So here is the basically artery contain a deoxygenated blood and carry the oxygen from the blood vessel during the breathing from the air sac to the blood vessel, so that move the to back into the heart. So that circulation is the pulmonary circulation, and systemic circulation will be occur. While here we will discuss just first of all that is the alveolar sac. This alveolar sac is contained inside is the mucous membrane like structure that is known as the surfactant. So surfactant is made up of 90% is water, as well as the innate immune system, which that means the macrophage and dendritic cell are present. So it is also the cell 82 lung epithelial cell is present. While the first of all we will discuss about the indication of the coronavirus infection. So the shortening of breathing, fatigue, nausea, fever, germ attack due to the loss of surfactant, and heart failure. So that's cause. So that is the symptom to understand is a physically, but when we will discuss about the check of the coronavirus PCR, we will discuss in the next video. So here is the blood vessel. This blood vessel is basically contain a deoxygenated blood, for example, which that move into the alveolar sac, and that's move of the alveolar sac to exhale into the environment, while the oxygen will move inside to move into the tissue, and tissue use oxygen for Krebs cycle to produce energy. So that is the cellular respiration and the physical respiration, which that is the breathing, while the cellular is the Krebs cycle. So that is important to understand. So this coronavirus when infect into the and go into the alveolar sac, for example, this coronavirus is going into the alveolar sac. So the alveolar sac contain a many type of cell. So also contain a macrophage, lymphocyte, as well as the innate and adaptive immune cell is present. So innate immunity first of all we will discuss. So that's airspace, basically that is inside is the airspace. So the surfactant is surrounded by the epithelial cell. We will also understand what is the surfactant and their important role to breathing. So here the airway, epithelium, and submucosal gland are present, which that produce a mucus, and a mixture of bronchial and alveolar surfactant, which that is mixed to each other, which that is the near to the submucosal gland as well as downward, so into the alveolar sac. So here is the basically so here is the inhibitor of the drug to prevent infection site by the binding of the ACE2 receptor by the spike protein is the hydroxychloroquine. So that is important, and which that is stop the infection site and stop to the spike protein of the coronavirus. So that means if we will not discuss, so that is the basically uh, without drug, that is infection, and that produce the replication into the into the AC2 lung epithelial cell, and after the replication to release this virus. Anyhow, during the releasing of the virus, we can also use that drug is known as the leflunomide. So this leflunomide is also used for it. So the here we will discuss about step by step to understand the infection and their physiology. So here the first of all receptor mediated viral entry. First of all, viral virus will be entered by the receptor. Receptor is the ACE2, while the second one is the viral replication inside of the cell. The cell is the ACE2 lung epithelial cell. So that's important thing to know about. These cells produce the surfactant. So the surfactant is important for breathing. Without surfactant, the breathing is a shortness of breath and not clear the breathing mechanism. So that is basically viral replication and immune cell activation. This immune cell activation is the first of all macrophage will be activated and produce the interleukin 6, MCP1, interferon gamma, and interleukin 1 beta. This is the recruit to the more macrophage into the from the blood vessel, as well as recruitment to the of the monocyte. So this monocyte is the precursor of the macrophage after the proliferation of this monocyte to become a macrophage. So that is the future macrophage monocyte, while the recruitment of the more macrophage, as well as the natural killer cell and neutrophil and B cell recruitment. How to B cell, that is the macrophage recruitment, monocyte, macrophage, and neutrophil, which that is the basically innate immune system, which that is adapted during the birth, while the adaptive immune system is here is the lymphocyte. This T helper cell which they recognize the antigen, and after they recognize to antigen to produce this chemical interleukin 2, interleukin 6, interleukin 12, interferon gamma, which that is recruit the more macrophage as well as B cell. B lymphocyte is very important to produce the antibodies, and that is proliferate into the plasma cell as well as the B memory cell. So the B memory cell is used for the next infection to quick release the antibody, while the plasma cell is at the time of infection, which that is cover that situation. So that's the plasma cell and B cell is interact each other for uh, adaptive immunity. So remember, lymphocyte is important for the adaptive immunity, while on the other cell is used for the basically innate immune system, which that is used for the quick mechanism to uh, endocytose the antigen from the dendritic cell as well as macrophage. So here we will discuss about the simplified view of lung surfactant action in an alveolus. So the surfactant is important. Without surfactant, it's nothing and not breathing. So here I am drawing the structure of that is the alveolar sac, which that contain mucous membrane. It is known as the surfactant. So surfactants contain antimicrobial activity because it's contain macrophage as well as the dendritic cell, which they recognize the antigen to give the response by the different cascade of the cell to proliferate and produce the antibodies. So here inside is the fibrinogen. So you can see that the yellow color shows that is the club cell or Clara cell, while the airway epithelium is present in the upper neck region of the alveolar sac, and submucosal gland is present basically, which that produce the mucosa, mucous membrane like structure, while here is the region of the neck and alveolar sac. Between this admixture of alveolar and bronchial surfactant, which that is important, and that is trapping more things. So important surfactant is to reduce and increase the volume of the alveolar sac. We will discuss in next. So here is the suboptimal surfactant fluid is present in the downside. There is important thing is decrease ability to inflate. First, ability of the inflating of that air sac is due to the loss of surfactant, which that is not allowed to decrease the volume inside. So the alveolar damage high ACE type 2 turnover. So it means the ACE2 type 2 cell which that contain telomere which that produce a surfactant is lost due to the infection of the virus which that damage this cell. So the ACE type 2 telomere shortening and senescence, it means that shedding of this telomere due to the infection. So this infection area which that is caused the loss of surfactant. So the meaning of loss of surfactant is important to understand. When alveolar radius smaller, first of all, alveolar radius smaller, we will discuss about the concept. So the alveolar radius will be smaller, it means the breathe out. It means the carbon dioxide and some oxygen molecule will be breathed out completely due to the surfactant. Surfactant decrease the volume of the alveolar sac during the breathe out exhalation, and surfactant film compressed and surface tension is smaller and complete exhalation, while when alveolar radius is larger, it means the inhalation time and the surfactant film expand like that and surface tension is larger. It means the pressure will be down while during the contraction the pressure will be rise. And important thing is that loss of surfactant due to the infection of that virus and less carbon dioxide move out due to high volume. So the less carbon dioxide move out, it means the carbon dioxide remain inside. So that is why the shortening of breathing, because again and again breathe for consumption of more oxygen, but the carbon dioxide not completely exhale. So that's why. And so important thing is to understand is the Laplace's law to understand that is the breathing mechanism, and that's volume we will discuss. So here is the change of the variable of the pressure and the 2T, T means the tension of the surfactant and our radius, which that is given the P is the pressure and T is the tension and R the radius. For example, the tension will be decreased due to the surfactant. It means the pressure will be also decreased. It means it's not too much, a partial pressure will be decreased, while if we will talk about the decrease of the partial pressure, so it means the radius will be increased. So thanks for watching.